we will discuss in this class about mitosis cell division. Now let's talk about the concept of chromosome. Chromosome is a thread-like object made up of chromatin. Actually, this chromatin is made up of DNA and special type of structural protein called histone. And according to the string and bed model, chromatin is present in the chromosome is present in the nucleus of the cell, and the size, the length of chromosome is thousands times than that of the dimension of nucleus. So it was coiled like a snake, and the chromosomes are visible in microscope under light microscope during metaphase stage of cell division. So this is cell, nucleus, inside nucleus there are chromosomes. Inside chromosome there are DNA. Now, what happens once parts of chromosome? Mainly three parts: telomere, that is the chromosome tips, this one; centromere, middle region where the spindle fiber attach; and chromatid, this one or this one. Chromatid in monovalent chromosome there are one chromatid, but in this one monovalent chromosome, and in bivalent chromosome, this is bivalent chromosome, there are two sister chromatids. And from monovalent chromosome to bivalent chromosome, this step occurs in the interphase stage, the longest stage. This is the diagram of monovalent and bivalent chromosome. Monovalent chromosome. This is monovalent chromosome. This is one chromosome with one chromatid. DNA copying of after copying of DNA, there is one chromosome and two sister chromatids. These chromatids are attached by cohesive force. Now the chromosome, during chromosome condensation, these chromatids bends, and so there are one chromosome with two sister chromosomes, and after the chromosome separation there leads to two chromosomes with one chromatid. Phases of mitosis cell division. There are 5 phases. First one interphase, then prophase, then metaphase, then anaphase, and next the last one is telophase along with cytokinesis. Interphase, here the replication of DNA strand and formation of two sister chromatids joined at the centromere, and this takes place the longest time. The next one. Early prophase, here chromosomes begins moving apart, and nuclear envelope. nuclear envelope. Nuclear envelope fragment undergo fragmentation and nucleolus starts disappearing. Late prophage where spindle fiber is formed and centrosome of the chromosome, the middle part of the chromosome, is attached to spindle fiber, sometimes called electromeric or kinetochore fiber. Chromosomes has no particular in this stage. Chromosome has no particular orientation. Metaphase. Fully formed spindle fiber consists of folds, esters and fibers. Chromosome attached to centromeric spindle fiber line up at the metaphase plate, and spolar spindle fiber reaches beyond the metaphase plate and overlap. Anaphase. Daughter chromosome having one centrosome and one single chromatid moves towards the pole as centromeric spindle fiber disassembled and pull daughter chromosome towards pole. Telophase. Their chromosome begins to disperse into chromatin and nuclear envelope reforms around each cluster of chromosomes and spindle begins to break apart. Nucleolus becomes visible and the cytokinesis occurs at the same time to form two identical cells. What happens when cell division what happens when cell division goes wrong? First one, there are three diseases. One, Down syndrome, sometimes called trisomy 21. Here an extra copy of chromosome 21 instead of two homologous chromosomes. They have three chromosomes. Triple X syndrome, an extra copy of X chromosome in female somatic cell. XYY syndrome, an extra copy of Y chromosome in male somatic cell. Thank you for watching.